A previous pope wrote the shroud was, quote, a truly mysterious image which no human artistry was capable of producing. Lara, but we have the book, the broadcast, and now the app.
He was wrapped in linen, and then his body was put in the tomb.
What if the world's most controversial artifact finally revealed its secret through science? For centuries, the Shroud of Turin has carried the haunting image of a crucified man, defying any known explanation. Scientists have tested it for decades, trying to prove it false, but new DNA evidence has changed everything. Hidden inside ancient fibers, researchers discovered biological material from multiple continents, pointing to a timeline that refuses to fit neatly together. The mystery question is no longer what the shroud is, but who left that image behind. The photograph that changed everything. For skeptics, the Shroud of Turin has historically told a remarkably simple story. It was merely one of history's most convincing religious forgeries. Over the course of centuries, alternative theories multiplied rapidly. Some historians pointed directly to Leonardo da Vinci, while others suspected that a highly skilled medieval artisan created the artifact specifically to inspire pilgrims and generate massive church revenue. While devout believers saw a deeply sacred relic, hardcore skeptics saw nothing more than a clever fabrication. For 600 years, both sides relied almost entirely on personal faith or cynical suspicion, with very little hard physical evidence available to settle the debate. But that dynamic changed completely with the advent of modern technology. Researchers from the diverse fields of genetics, forensic pathology, materials science, spectroscopy, and high-energy physics began treating the shroud as tangible physical evidence rather than just a symbolic religious object. They carefully extracted ancient DNA from the linen fibers. They applied advanced digital imaging and sophisticated X-ray analysis to the cloth. They even studied the embedded bloodstains at a strict molecular level. Consequently, the ancient linen was transformed into a scientific archive where every single fiber, stain, and microscopic particle potentially preserved traces of its long, mysterious history. The resulting data rarely offered simple answers. Genetic analysis suggested direct contact with diverse people from multiple distinct geographic regions. Intensive forensic examinations identified human blood consistent with severe physical trauma. Advanced imaging revealed optical characteristics unlike any known painting technique on Earth. Furthermore, material analyses continually challenged long-held archaeological assumptions. As a direct result of these findings, the investigation quickly became far more complex than a simple binary authenticity debate. But long before DNA sequencing, forensic chemistry, or advanced digital imaging transformed the modern discussion, the shroud's modern scientific journey began with a single photograph taken in 1898. This pivotal moment occurred when Secondo Pia, an Italian lawyer and passionate amateur photographer, captured the first clear images of the cloth. When he later developed the glass negatives, he was absolutely stunned. The faint, shadowy figure on the linen suddenly appeared as a highly detailed, positive image of a crucified man. For the very first time in history, people could clearly witness the wounds, the distinct blood flows, and the haunting, realistic facial features. That landmark 1898 photograph radically shifted the cultural conversation. What critics had previously dismissed as a crude medieval painting suddenly looked far more sophisticated. The image on the cloth behaved exactly like a photographic negative. It contained unique three-dimensional information and displayed anatomical details that no artist of the Middle Ages could have produced with the rudimentary materials and limited anatomical knowledge available at the time. This single photographic event sparked serious, lasting scientific interest. It encouraged independent researchers to examine the shroud not merely as a religious curiosity, but as one of history's most intriguing forensic mysteries. From that specific point forward, the linen would face decades of rigorous testing across multiple academic disciplines. Microscopic pollen grains trapped in the weave were later cataloged to reconstruct possible ancient travel routes. Biological traces were carefully sequenced to identify the distinct populations that might have handled it. Blood chemistry was analyzed to understand the signs of extreme physiological stress. Additionally, Forensic specialists meticulously studied the wound patterns, which were remarkably consistent with the historical realities of Roman crucifixion. Each distinct field contributed another vital piece to an increasingly complex historical puzzle. The image itself continued to defy any easy scientific explanation. It appeared strictly confined to the outermost layer of the fibers. It showed no detectable signs of pigments, binders, or traditional brushstrokes. It carried unique optical data that modern digital analysis easily converted into a three-dimensional relief map. This specific characteristic sets the cloth completely apart from every known artistic technique. Yet before any of these advanced laboratory studies took place, that 1898 photograph planted a simple but exceptionally powerful question in the minds of scientists and historians alike. What exactly are we looking at? DNA, a biological archive. The Shroud's greatest secrets were never actually visible to the naked human eye. Instead, they remained completely hidden within microscopic traces of dust, pollen, and organic material trapped deeply between the linen fibers, serving as silent witnesses to centuries of travel, handling, and environmental exposure. Every single person who touched the cloth, every geographic place it passed through, and every distinct environment it encountered left behind a unique biological fingerprint. In 2015, researchers from the University of Padua set out to systematically examine those hidden microscopic traces. Their primary goal was not to prove a miraculous or supernatural origin, as that falls completely outside the scope of modern genetics. Instead, they wanted to meticulously reconstruct the cloth's long historical journey, where it had traveled, what specific environments it had encountered, and which human populations may have come into direct contact with it over the course of centuries. To successfully recover this fragile evidence, the scientific team utilized sterile micro-vacuum devices fitted with ultrafine filters. These specialized tools carefully extracted dust, pollen, loose fibers, and organic debris, not just from the outer surface, but also from the narrow, tight spaces between the woven threads, where ancient material can remain safely protected for remarkably long periods. They also thoroughly studied samples that had been carefully preserved from the Shroud's major 1998 restoration project. The extraction process required an extraordinary level of caution. Ancient DNA is extremely fragile, and even a single modern skin cell or a stray human breath can easily overwhelm and contaminate centuries-old genetic material. Because of this risk, every step of the laboratory protocol was specifically designed to minimize contamination. Once successfully collected, the material was immediately taken to specialized clean laboratories for next-generation sequencing, a highly powerful technology capable of analyzing millions of distinct DNA fragments simultaneously. The researchers focused primarily on mitochondrial DNA, which naturally occurs in far greater quantities than nuclear DNA and survives significantly longer in ancient samples. Because mitochondrial DNA is passed down strictly through the maternal line and shows distinct geographic patterns, it offers highly valuable clues about ancestry, regional migration routes, and historical population movements. Powerful computing systems then process these millions of genetic sequences, comparing them directly against international reference databases covering diverse populations from Europe, the Middle East, Africa, and Asia. The ultimate aim was entirely straightforward: determine whether the biological traces pointed to a single clear geographic origin or revealed a far more complex history. The final results were far more complicated than anyone had expected. Instead of showing the clear genetic signature of a single individual or a localized population, the analysis identified DNA associated with multiple diverse ancestral lineages. The findings, officially published in the journal Scientific Reports, strongly suggested that the cloth had accumulated biological material from people originating in several entirely different regions of the world. This discovery immediately challenged any simple explanation. If the Shroud had remained in one single location for most of its history, researchers would have expected a much more localized, uniform genetic profile. Instead, The physical evidence pointed to centuries of continuous contact with diverse populations, a finding highly consistent with an object that had been transported, displayed, handled, and venerated across different cultures and vast geographical regions. Among the most notable findings were specific mitochondrial haplogroups commonly associated with the Middle East, including lineages found today in present-day Israel, Jordan, Lebanon, and Syria. While these genetic signatures cannot definitively prove who originally owned the cloth, they align perfectly with the shroud's traditional historical narrative. The analysis also revealed strong European genetic lineages, as expected given the shroud's well-documented historical presence in France and Italy since the mid-14th century. It was routinely handled by clergy, nobility, textile conservators, and countless religious pilgrims over hundreds of years, leaving a substantial, undeniable European genetic imprint behind. More surprising, however, were distinct traces linked directly to North and East Africa, including specific lineages associated with ancient Christian communities in Egypt and Ethiopia. Additional genetic signatures pointed to the Indian subcontinent and even East Asia. Taken together, the sequenced DNA reflected biological traces spanning Europe, the Middle East, Africa, and parts of Asia. Rather than a single geographic fingerprint, the ancient linen carried a remarkably diverse record of human contact. How exactly could one piece of cloth accumulate such a wide range of genetic material? The most plausible answer lies in its long, complex journey through centuries. If the shroud passed through major ancient population centers, was displayed publicly, and was handled by pilgrims and travelers from many different regions, it would naturally collect biological traces from countless individuals over time. Some scholars link this immense genetic diversity to the historical tradition of the Mandylion, an image of Christ described in Byzantine and Syriac sources, said to have been kept in Edessa for a long period. The city of Edessa sat at a highly strategic point along ancient trade routes linking the Mediterranean world with Persia, Arabia, India, and eventually China via the famous Silk Road. If the relic was indeed housed there, repeated public exposure would have created many opportunities for diverse biological material to accumulate on the fabric. Later, the cloth is believed to have reached Constantinople, one of the most cosmopolitan cities of the medieval world, further increasing its contact with people from Europe, North Africa, and Asia. After completely disappearing during the sack of the city in the Fourth Crusade in 1204, it eventually reappeared in France in the mid-14th century, before ultimately reaching its current resting place in Turin. This proposed historical route offers a highly reasonable explanation for the genetic diversity found in the linen today, while some researchers caution that extensive handling over centuries makes it incredibly difficult to separate ancient traces from modern contamination, the DNA. Evidence still paints a clear picture of a remarkably complex biological history, one that simply cannot be reduced to a single place, period, or population. Human DNA, however, was only one layer of the puzzle. The linen had also preserved another vital category of microscopic evidence that came not from people, but from the diverse plants that surrounded it during its long journey. Pollen and botanical evidence. Pollen analysis, known as palynology, added another critical dimension to the investigation. Unlike DNA, which reveals information about the people who contacted an object, pollen preserves clues about the environments through which the cloth passed. Pollen grains possess durable outer walls that can survive for centuries, serving as microscopic records of geographic origin and historical movement. Several researchers examined the shroud from this perspective. The most notable were Swiss forensic scientist Max Frey, a pioneer in forensic pollen analysis, and later, Israeli botanist Avinoam Danian of the Hebrew University of Jerusalem. Working independently, both analyzed pollen recovered from the linen using adhesive sampling techniques that lifted particles embedded deep within the fabric's fibers. Their findings revealed pollen from dozens of different plant species. Many were entirely consistent with the shroud's well-documented history in Europe. Approximately 17 species native to the continent were identified, an expected result, given that the relic has spent centuries in France and Italy and has been repeatedly displayed, transported, and handled. The remaining pollen attracted far greater attention. Researchers identified numerous species associated with the Middle East, Anatolia, and regions corresponding to the historical route proposed for the shroud through Edessa and Constantinople. While pollen alone cannot establish where the cloth originated, it provides valuable environmental clues that complement archaeological and historical evidence. Among the most discussed discoveries was pollen attributed to Gundelia tournefortii, a thorny flowering plant native to parts of Israel and the surrounding region. According to the published analyses, this species represented an unusually large proportion of the pollen recovered from the linen. This prompted researchers to investigate why a plant with such a restricted natural habitat appeared so prominently on the cloth. The significance extends beyond geographic distribution. Gundelia tournefortii grows throughout the hills surrounding Jerusalem and typically flowers during early spring, closely corresponding with the period of Passover described in the New Testament accounts of the crucifixion. Because of its long, rigid thorns, some researchers have proposed that it may resemble the vegetation that could be fashioned into the crown of thorns described in the Gospel narratives. That interpretation remains speculative, but it has become one of the more widely discussed aspects of the botanical research. Researchers also identified pollen attributed to Zygophyllum dumosum, another species whose natural range is largely confined to the Judean desert and parts of the Sinai Peninsula. The presence of multiple plant species in the same geographic region strengthened the argument that the botanical evidence warranted serious consideration. Together, the pollen studies presented a picture that was difficult to ignore. Instead of reflecting only the vegetation of medieval Europe, the botanical evidence included species associated with landscapes much farther east. Supporters argue this pattern aligns with the shroud's traditionally proposed journey through the Middle East before arriving in Europe. Skeptics note that pollen identification can be challenging, and that some of Max Fry's conclusions have been questioned by later researchers, making the evidence suggestive rather than definitive. Even so, the botanical findings remain an important part of the broader investigation. Unlike paint or pigments, pollen accumulates naturally through environmental exposure and becomes trapped within fibers over time. Whether interpreted as evidence of an ancient journey or of centuries later contact, the microscopic grains preserved on the linen add another layer to the shroud's remarkably complex history. The pollen told one story, but another cloth, quietly preserved in Spain, would soon add an unexpected twist to the investigation. The Sudarium of Oviedo, the shroud's forgotten companion. Just when it seemed scientists had reached the limits of what the shroud could reveal, their attention shifted to another artifact, quietly preserved in northern Spain for more than 1,000 years. The Sudarium of Oviedo is a small linen cloth that bears no facial image. At first glance, it appears far less remarkable than the shroud. It is stained, worn, and marked only by patches of dried blood and bodily fluids. Yet some researchers believe it holds one of the strongest pieces of forensic evidence in the entire debate.
The shroud is not the only burial relic of our Lord.
Right.
The other big one, is the Sudarium of Oviedo.
Yes.
And this is the head cloth.
According to first-century Jewish burial customs, a separate cloth was often placed over the deceased's face before the body was wrapped in larger burial linen. The Gospel of John describes exactly this: 2 different cloths inside Jesus' tomb, one wrapped around the body and another covering the head. If the shroud is the larger burial cloth, could the Sudarium be the second? Rather than searching for images or miraculous impressions, forensic investigators focused on something more ordinary: the blood stains. Every stain tells a story. The direction of flow, the angle at which it dried, and the amount absorbed into the fabric can reveal how a body was positioned when the blood was deposited. Researchers carefully mapped the blood patterns on both cloths. They compared the location of stains around the nose, mouth, and beard area, along with evidence of fluid that flowed after death. Several independent studies reported striking similarities, suggesting both cloths may have covered the same wounded face at different stages of burial. Investigators also concluded that the injuries reflected on both linens were consistent with a man who had suffered severe facial trauma and crucifixion. The head appeared to have been positioned upright for some time before being laid horizontally. If accurate, the blood patterns on one cloth would naturally complement those found on the other. Supporters argue that these independent details are difficult to dismiss as coincidences. The sudarium carries no full-body image and developed an entirely different pattern of stains. Yet, despite their differences, both cloths share several forensic characteristics, including blood types consistent with type A in some analyses. Critics point out that ancient blood evidence is notoriously difficult to interpret. Some question whether stain comparisons alone can establish that two cloths wrap the same individual. Others argue that centuries of handling and environmental exposure make definitive conclusions nearly impossible. Even so, the sudarium adds another important layer to the mystery. Instead of relying on a single relic, researchers suddenly had two separate pieces of linen that some believe tell parts of the same story. Whether they truly belong to the same man remains a matter of debate, but together they raise a compelling question. If two independent burial cloths preserve compatible forensic evidence, what other traces might still be hidden within the fibers of the shroud itself? The answer, it turned out, was in the blood. Blood, trauma, and crucifixion. The blood on the shroud was not paint, it was human. Some analyses identified it as type AB, while others identified it as type A. Most importantly, it contained markers that told a story of extreme suffering. A team of Italian researchers led by Professor Giulio Fanti of the University of Padua, working alongside physicians from a hospital in Trieste, re-examined the stains using advanced techniques, including transmission electron microscopy and Raman spectroscopy. Analyzing the material at the nanoscale, they discovered something very different from pigment. The scientists found not paint, but actual human blood, identified as type A in their study. While rare today, this blood type has also been documented in other ancient Christian relics, including the sudarium of Oviedo. This was not normal blood. It contained elevated markers, including creatinine and ferritin bound to hemoglobin. These compounds indicate extreme physical trauma, severe stress, dehydration, and massive muscle breakdown. In conditions like rhabdomyolysis, the body releases large amounts of creatinine into the bloodstream. The presence of these substances suggests the individual suffered intense and repeated physical violence before death. This evidence aligns with historical accounts of Roman scourging. The Romans used a flagrum, a whip embedded with pieces of bone or metal. Forensic interpretations suggest the pattern is consistent with more than 100 blows. The chemistry points to a level of suffering that cannot be recreated by paint or artistic technique. It includes signs of shock and potential organ failure. The color of the stains raised another important question. Fresh human blood is typically bright red. As it ages, it gradually darkens to a deep brown due to chemical changes in hemoglobin. The stains on the shroud, however, have retained a reddish appearance that many researchers consider unusual for blood believed to be centuries old. One proposed explanation centers on bilirubin, a yellow-orange pigment released when red blood cells break down. Individuals who experience severe physical trauma often develop unusually high levels of bilirubin in their bloodstream. Some forensic investigators suggest that a combination of bilirubin and oxidized hemoglobin may have helped preserve the blood's reddish coloration for far longer than expected. While this hypothesis remains the subject of scientific discussion, it offers one possible explanation for a feature that has puzzled researchers for decades. The blood raised one compelling question, but the age of the linen itself raised another, far more controversial one. The radiocarbon dating debate and new methods. In 1988, 3 independent laboratories at Oxford, Zurich, and the University of Arizona performed radiocarbon dating on samples taken from the shroud. Their results placed the fabric between 1260 and 1390, leading many researchers to conclude that the relic was a medieval creation. The debate did not end there. Over time, several researchers questioned whether the tested sample accurately represented the entire cloth. The material analyzed came from a corner that had been heavily handled during public exhibitions and may have undergone medieval repairs following damage in earlier centuries. Among the most prominent critics was chemist Raymond Rogers, a member of the Shroud of Turin research project. After examining fibers from the same region used for radiocarbon dating, Rogers argued that the sample differed chemically from the rest of the linen. His analysis suggested the presence of cotton fibers, dye, and other characteristics consistent with later repairs, rather than the original fabric. If that interpretation is correct, the radiocarbon measurements may have reflected the age of a repaired section, rather than the cloth as a whole. Other researchers continue to dispute this conclusion, maintaining that the 1988 dating remains valid. The question of whether the tested sample accurately represented the entire shroud remains one of the most actively debated issues in the artifact's scientific history. The controversy encouraged scientists to explore dating techniques less reliant on surface contamination and more focused on the linen's internal structure. One recent approach was introduced in 2022 by physicist Liberato De Caro and his colleagues using wide-angle X-ray scattering, or WAXS. Rather than measuring radioactive decay, WAXS examines the microscopic structure of cellulose within linen fibers. As flax ages, its molecular organization gradually changes due to exposure to heat, humidity, and other environmental conditions, creating patterns that can be compared with those of textiles of known age. When De Caro's team compared the shroud's fibers with ancient and medieval linen samples, they concluded the fabric appeared more consistent with considerably older textiles, including examples recovered from first-century archaeological sites, such as Masada in Israel. The study proposed that, under appropriate preservation conditions, the linen could be substantially older than the medieval date suggested by radiocarbon testing. Not all researchers agree with that interpretation, and WAXS has not replaced radiocarbon dating as the accepted standard for determining the age of archaeological textiles. Even so, the study reopened a debate that many considered settled, reminding both supporters and skeptics that the shroud continues to resist simple conclusions. With so much evidence gathered over the decades, one question remains: why won't the Vatican just declare it authentic? The Vatican's position in real life. After decades of scientific studies, one obvious question persists. If so much evidence continues to fuel debate, why has the Vatican never simply declared the Shroud of Turin to be the authentic burial cloth of Jesus? The answer is more complicated than many realize. Contrary to popular belief, the Catholic Church has never officially declared the shroud authentic, nor has it declared it a forgery. Instead, the Church has deliberately remained neutral, choosing to separate questions of scientific evidence from matters of faith. This position surprises many people because the shroud has been displayed publicly by several popes and venerated by millions of pilgrims. But for the Vatican, veneration is not the same as authentication. Over the years, different popes have spoken about the relic with deep respect, while carefully avoiding definitive historical claims. In 1998, Pope John Paul II described the shroud as a mirror of the gospel. He encouraged scientific research while reminding believers that faith does not depend on relics. Years later, Pope Benedict XVI referred to it as an extraordinary icon, reflecting the suffering described in Christ's Passion. Pope Francis has also treated the cloth as a powerful object of devotion, without declaring it the actual burial linen of Jesus. Why such caution? Authenticating a relic is fundamentally different from studying one. Science can examine linen fibers, pollen grains, blood chemistry, and microscopic DNA fragments. It can estimate age, identify contaminants, and compare forensic patterns. But proving that a particular cloth once wrapped Jesus of Nazareth requires historical certainty that science alone cannot provide. The Vatican also understands that the evidence remains contested. While some researchers argue the combined findings point toward a first-century origin, others support a medieval date or question the interpretation of key studies. Declaring certainty while experts remain divided would place the Church in the middle of an ongoing scientific debate, Instead, the Vatican has largely welcomed responsible investigation. Researchers have been granted access on several occasions, and the Church has generally encouraged careful examination rather than discouraging it. Its position has remained remarkably consistent. Wherever the evidence leads, the Shroud should inspire reflection, not replace faith. Ironically, this cautious approach has only fueled the mystery. Every discovery, whether DNA, blood chemistry, or advanced imaging, raises the same question. If the Vatican refuses to make a definitive judgment, could science ever do what the Church has deliberately chosen not to? The image that defies explanation. Despite decades of scientific investigation, the Shroud's central mystery remains unresolved. The image is confined to an extraordinarily thin layer on the outermost fibers, measuring only a few hundred nanometers in depth. It contains no evidence of pigments, ink, or brushstrokes. Instead of behaving like a conventional work of art, the discoloration appears to result from a chemical alteration of the fibers themselves. How that alteration occurred remains unknown. Researchers have proposed explanations ranging from natural chemical reactions to more unconventional hypotheses involving brief bursts of radiant energy. None has achieved a broad scientific consensus. No laboratory has successfully reproduced every known characteristic of the image using a single process. Further studies have only deepened the mystery. Digital analyses suggest the image contains three-dimensional information. Variations in intensity correspond to the distance between the body and the cloth, unlike conventional paintings or photographs. Other investigations have reported evidence consistent with Middle Eastern pollen, mineral particles similar to those found in the Jerusalem region, bloodstains associated with severe physical trauma, and microscopic features that continue to challenge straightforward explanations. Taken individually, none of these findings settles the debate. Taken together, they have transformed the Shroud from a religious relic into one of the most extensively studied archaeological objects in the world. Genetics, forensic pathology, chemistry, physics, botany, and materials, science have all contributed important pieces to the puzzle. Yet no single discipline has provided a complete answer. Some researchers believe the cumulative evidence is consistent with a much older origin than previously thought. Others argue that contamination, incomplete historical records, and unresolved methodological questions make definitive conclusions impossible. Neither position has closed the case. The Shroud of Turin remains exactly what it has been for centuries, a silent piece of linen that continues to invite investigation. Whether future discoveries ultimately identify it as the authentic burial cloth of Jesus, an extraordinary medieval creation, or something else entirely, one fact is difficult to dispute. More than 600 years after it first entered the historical record, the Shroud still refuses to surrender all of its secrets. Could the Shroud ever be proven? After more than a century of scientific investigation, billions of microscopic fibers examined, and countless studies published, one question still refuses to disappear. Could the Shroud of Turin ever actually be proven to be the burial cloth of Jesus? At first, the answer might seem simple. Just keep testing it. Develop better technology, gather more evidence. Eventually, science will settle the debate once and for all. But the reality is far more complicated. Science is exceptionally powerful at answering questions about what something is. It can determine the composition of linen fibers, identify traces of blood, detect ancient pollen, recover DNA fragments, and estimate the age of a material. Each discovery adds another piece to the puzzle. Yet, proving who a cloth belonged to is an entirely different challenge. There is no verified sample of Jesus' DNA to compare against. There is no historical record that can trace the linen continuously from a first-century tomb to the modern day. Even if every future test pointed toward an origin in ancient Jerusalem, that alone could never demonstrate beyond doubt that the cloth wrapped Jesus of Nazareth and no one else. This is why the debate has endured for generations. Supporters see multiple lines of evidence converging toward the same conclusion. Skeptics argue that extraordinary claims require extraordinary evidence, and they point to uncertainties surrounding dating methods, contamination, and interpretation. Both sides are often looking at the same data, but asking fundamentally different questions. Ironically, every technological breakthrough has made the mystery more complex rather than less. Photography revealed an image unlike ordinary artwork. Forensic analysis uncovered signs of severe physical trauma. DNA studies suggested a remarkably long and complicated history. Advanced imaging exposed details invisible to the naked eye. Yet none of these discoveries has produced the single piece of evidence capable of ending the debate. Perhaps that is because the Shroud occupies a unique place where history, archaeology, religion, and science overlap. Each discipline offers valuable insights, but none can answer every question on its own. For some, the Shroud will always remain a sacred object of faith. For others, it is an extraordinary archaeological mystery. For many scientists, it stands as one of the most fascinating unsolved forensic puzzles ever examined. So, could the Shroud ever truly be proven? Maybe. Future technologies may reveal details that today's instruments cannot detect. Or perhaps the decisive evidence has already been lost to centuries of time, handling, fires, and restoration. Until then, the linen continues to challenge everyone who studies it, not because it provides all the answers, but because every attempt to solve its mystery seems to uncover another question waiting beneath the surface. Thanks for watching. Now check out the videos popping up on screen for more unbelievable stories.